Thanks so much, Mary. It's a pleasure to be here. And I just wanted to give a few brief slides to sort of give you an indication of how cancer genomics is being applied to precision medicine. So we've known for some very long time, actually, over 100 years, that cancer is a disease of the genome. This was first postulated in the early 1900s, but really came to fruition with the efforts of pathologists such as Dr. Janet Rowley, shown here, who early in her career began looking at cancer cell chromosomes, under the microscope, and was able to sort of discern that patients with the same diagnosis actually had very similar looking chromosomes, and these were translocations that were later shown to force together two proteins that were never meant to be together, and these were cancer drivers, as we commonly refer to them today. So microscopy has led the field for some very long time, and yet what has come into play, especially since the advent of the Human Genome Project, which I was fortunate enough to participate in early in my career, and the advent of new technologies such as next-generation sequencing, which is shown here, a prototypical box that you do next-generation sequencing on, has really changed our understanding of cancer as a disease of the genome. So what we can do right now today on an instrument that is shown here that costs on the order of about $1 million is sequence the genomes of several cancer patients literally overnight and for a very low cost compared to when we sequenced the first cancer genome in 2008, which was a $1 million project. So essentially we're doing the same thing as pathologists, it's just that our microscope has changed a bit. We're not trying to replace pathologists, so I don't want anyone in the room who's a pathologist to worry. What we're really doing is we're adding detail to what you already see under the microscope. So rather than seeing a cancer chromosome, if you will, with a known translocation such as BCR-ABL or PML-RAR alpha, like or T1517 rather, like Janet first discovered under her microscope, we're able to sequence the genome and piece back together, in particular, the cancer genome and the normal genome of an individual, and have a very intimate comparison of what's different about that cancer cell's DNA versus the normal DNA from that individual. And as I'll walk you through in the next few slides with a few examples from lung cancers, this has dramatically changed the way that we think about, diagnose, and ultimately treat cancers today. So old school lung cancer diagnosis by microscope essentially took a section from a biopsy needle, for example, did prototypical hematoxyl and eosin staining, and then under the microscope was diagnosed as squamous cell disease, large cell lung cancer. These are the minority of patients, and the majority of patients have lung adenocarcinoma. And as you can see, regardless really of the designation under the microscope, the outcome was the following. You had surgery, radiation, and chemotherapy, and hope for the best. So how has this really changed? Well, in the era of targeted therapy, as shown here, and this was really work, as you can see from the citation on the bottom of the screen, done right as we were coming out of sequencing the human genome in the early 2000s. We were then able to identify where genes were located, go in and amplify them out by PCR and do Sanger sequencing, sort of old school as we consider it now today. But back then, in this study, working with Harold Varmus, who was then at Memorial Sloan Kettering, and putting patients with advanced lung cancer onto these very early tyrosine kinase inhibitors, we were able to compare the DNA from lung cancers that had or had not responded, and we were able to show that patients with metastatic EGFR mutated lung cancers were actually the ones that were largely responsive to these targeted therapies. So that was 2004. Now everyone knows that medicine is a very cautious practice and is slow to change, but over time, and really a little bit more than 10 years' time, these drugs, after being approved by the FDA and used in metastatic cancers, now have been approved in first-line therapy for cancer. So right now, today, this picture has changed dramatically, and we know that it's changed even more dramatically than that one example that I just illustrated, because now the old pie graph, which is shown on the far left, is a new pie graph, which is not only the tissue or cell type of origin, but also the different types of drivers that we can identify in those cancers. And you can see EGFR there, you can also see KRAS, which Kayvon talked about, but you can also see that there are a panoply of other drivers now, many of which have also targeted therapies that correspond to them. So the scenario now, lung adenocarcinoma diagnosis by genomics, goes as follows. We now take that tumor biopsy. We still examine it under the microscope to determine the pathology, but we take part of that biopsy material, isolate DNA from it, for example, and if there's an EGFR mutation, the outcome is that targeted therapy to EGFR is now utilized in first line, as I mentioned. We also know that EGFR and KRAS occurring together is not going to work for EGFR agonists, and so we now go back through the traditional standard of care, surgery, radiation, chemotherapy. And just as a newer example, because we've been talking a lot today about immunotherapies, if we show that this patient has a high mutational burden, the likelihood of response to checkpoint blockade therapy is quite high. And so we know there's a good correlation there. This patient may go on to the immunotherapeutic. So I'll just finish by saying that while this sounds wonderful, and it is, there are still challenges, as Mary alluded to, and I just wanted to list these out to finish off my presentation. So we can find alterations in each individual patient's cancer DNA. Not all patients have a sample that could be tested. I just saw a very exciting paper that came out this week in JAMA Oncology showing that even if you now test in the circulating blood, patients with very high tumor burden, you can pick up these mutations even if there's not a tumor biopsy to study, and many of these patients were put onto targeted therapy and showed a high response rate. So I think this is even changing. But the other part of that equation is, do you have access to genomic testing? And the answer for many patients is largely no. So we talked about disparities. This is certainly one. And the fact that most patients, about 80% of patients, get their cancer care in a community-based setting means that they are likely not be able to take advantage of the genomic testing. We've also talked a lot about cancer drugs that work, but the reality is that only about 20% of known cancer drivers are druggable. KRAS notwithstanding, there are plenty of others that just remain without targeted therapies. Another reality is that many drugs are developed to target the same gene, but not all drugs are equivalent with respect to their efficacy, and which drug you get depends on a lot of factors. We certainly don't understand all mutations, so next-generation sequencing has been fantastic in increasing our worldview of what drives cancers. But in reality, in the clinical setting, many of these mutations are what we call variants of unknown significance, or VUSs. We really don't understand whether they're actually changing the resulting protein to the point where the cancer is going to be responsive if they're drugged. And then lastly, as we've talked about a lot today, I think most patients treated with single targeted therapy develop drug resistance, so we have to start paying attention to the potential for combinations. Thanks very much.